A very good morning to you people. Our today's first lecture for this entire course is Comparative Genomics, Genetic Mapping, Physical Mapping, the Single Nucleotide Polymorphisms, the Express Sequence Text and the GSS, means Genome Survey Sequence. In the entire part, we will be discussing how different elements like SNPs, ESTs or the Genome Survey Sequence GSS have helped or complemented the genetic mapping and physical mapping to know the position of all the genes in a chromosome. Okay, but the question comes is why we need to do it? Why there's a need to map different genes on a chromosome? See, the story behind all this like comes up from evolution. Evolution, we know one species come rise from the other one. Okay, as the evolution term came up from Charles Darwin. Okay, there's a similarity between different species, which defines model organisms. Something which are more similar to humans are used for testing the drugs. People already know that mice or monkeys are used as like model organisms for testing the drugs, which are specially meant for humans. Okay, so it defines the model organisms. So, coming back to the annotation part, why the term annotation or giving predicting the function is important based on the evolution. See, the reason is currently the proteins database or the genome database is substantially large. Okay, so whenever this data is there, we don't know. So whenever, say, suppose some person finds a new gene in his lab, what he do? Rather than wasting money on predicting the function, getting the function in the lab again, experimentally determining the function in the lab, what he do, he get the gene sequenced. Okay, but before putting it in the data bank NCBA genome database or the protein database, what he do? He compare it with the known sequences by using blast method or many different methods. Okay, and then the server estimate the function. How it estimates? How the server estimates? Server estimates based on the similarity. So if the given sequence is more similar to say arginine kinase or DNA polymerase or RNA polymerase, then the server says that it is 79 or 80% similar to RNA polymerase. So it is likely to be a RNA polymerase, assigning some score. Okay, we'll be discussing later in the course. So this is the property of server. It is the concept is annotation. You are predicting the function. Annotation is much more a complex part, but this is a basic step to do it. Finding the similarity and then assigning the function. Okay. Here multiple experimental methods microarrays for expression, gene expression data are used. Okay, where exactly the protein is functioning, what is the exact function, and all those things. So the entire part proteomics includes only not only the identification and the quantification of proteins, but it is also the determination of their localization, modification. Localization means where exactly the protein is functioning in the cell. What is the exact post-transcriptional or post-translational modifications going on the proteins? How the protein is interacting with the other proteins, and what are the activities played? Based on all of this, defines the ultimate function, which is the key concept of annotation, as published in Stanfield's in Science 2001. So now you understand. To do all of these, we require the different databases to predict the activities. Okay. Now see Whenever we are doing all these predictions, we especially work out like comparative genomics. Key concept, like whenever you like gave you the example, you input a sequence, you check it against the known databases. We checked out that 80% similar. So how the system finds out 80% similar? It compare your sequences, suppose the first sequence, to all the database sequence by building an alignment, sequence alignment. The readers or the persons who are new to this, let me tell you that the alignment is nothing but like overlapping of all the sequences by putting gaps. Okay, so some sequence is bigger, some sequence is smaller. So the smaller sequence is aligned with a similar length by putting the gaps. Gaps are also kept in the bigger sequence to understand, make it better, so that the more similarity comes up. Okay, understand? So that the like pairwise similarity is substantially highest. You need to maximize that part, and you need to put the gaps. But you cannot put too many gaps also. It is just an optimal number of gaps. Okay, so there's a gap insertion penalty, air gap extension penalty, then that gives rise to an alignment. Now, if you align multiple sequences, this is termed as a multiple sequence alignment. Okay, now see the conserved part, something which is consensus here, CONS means consensus, conserved part. Here you see the highest amount of conservation means this part is substantially conserved in all these sequences. Conservation percentage is highest, see, like that. And where the gaps are there means that is not at all conserved. Okay, no such conservation exists. So like that you define the part. Okay, so it helps us that these positions are probably the regulatory elements. Okay, they are regulatory elements which are conserved in all the species, like chimp, rhesus, like blush baby, tissue, all of them. Okay, they are conserved in all the sequences, all the different species, means their function is conserved. Okay, so without doing anything in the lab, just the computer has told you the exact correlation between different sequences. This is entirely the magic. Okay, to move it further, understand that there are three categories of similarities whenever we are doing such an analysis. First is homologous, means whenever you are taking all the sequences, some sequences are having the same evolutionary relationship and same function. They are termed homologous because they are having some like evolutionary relationship to us, like humans and like monkeys. Okay, having some evolutionary connection. Somewhere down the line, we have evolved from like monkeys, so there is a connection. But no connection between mouse and human. Okay, or like a mouse and say snake. Then what do you say? They are orthologous because there is a similarity there, but evolutionary relationship is not there. Understand? So there's a species is changed. Similarity is there, but species changed. Termed as a speciation event in the evolution, but function is retained. Then there comes paralogs. Okay, paralogous. Here what happens? Like this, the gene duplication event happened. Gene duplication means within the sequence itself, within the species or body itself, that gene which was functioning in like a heart, okay, it is now present in legs also, doing some other function. But both the places, the function is different, but like a similarity is there. So high similarity is there, but function is different. This is termed as paralogous. Okay. So to map up all this to find the exact position of all of them, okay, on a genome, like genetic mapping and physical mapping are normally used. Okay. Genetic mapping means we use the linkage. Like whenever the crossover exists. Crossover means when you cross parent A with parent B, what all children are produced, offsprings. Okay, remember the genetics term, offsprings. Okay, what all like offsprings or the children are produced, what is their genotype, what is their genetic makeup. Based on their genetic makeup and the parents' genetic makeup, you try to estimate. These genes are always coming together, means they are linkage. Linkage is there. These genes are always getting separated, means the distance between them is far apart. So using this basic principle of linkage, you are trying to find out the approximate location of different genes on a chromosome. But remember that it is not exact order. It is just approximate map. Okay, that it should be like this. When it comes to physical mapping, it is like a quite very close to the actual part, but not exact. Reason is because it is impossible to find the exact position. We just try to correlate. Okay, and what are the units here? Either the base pair, kilo base pair, mega base pair, giga base pair, like that. Base pair means one base. Like a kilo base pair means 1000 bases distance. Mega base pair means the distance between two genes is order in the order of like 10 to 6 bases. Giga bases means in the distance between two genes is in the order of 10 to 9 bases. Okay, so all this helps us to find the location of two genes or multiple genes on a chromosome so that we know their positions. Okay, because besides annotation or knowing the function, their position also helps us to understand the complete like strategy of the protein function properly. Okay, let us move forward. See this genetic map as I told you. Something closer means they're always going together. Something far apart means they'll always get separated in the children. The crossover exists. They'll always get separated in the children. A, B, B. So if the two are very close, A and parent like gene A and gene B will go together. But if they are far apart, the crossover will exist and AB will come up. Why I am calling A and BB? Because our DNA is having double stranded. I am telling the gene on both the chromosomes, both the pairs. Okay. Moving forward. Now the concept is SNPs, single nucleotide polymorphism. Understand that SNPs in all like position changes at one only a single position, single base is changed. So in DNA, ATGCG is there, adenine, thymine, cytosine, guanine bases. In RNA, understand that those who are new to this course understand that in RNA, four bases are there, but thymine is not there. Uracil is there. Adenine, like U is uracil, okay. And C and G like cysteine, like cytosine and guanine remains as such. Okay. Now understand that all these three forms of an amino acid, like a pair of three, is termed as a codon in like protein. So three bases together define a codon amino acid. Okay, the codon is just the like representation of amino acid on like representation of amino acid in terms of characters. So CTT, if it is there, it will like form glutamic acid. Okay, so it changes to CAT. So glutamine is now changed to valine because of change of thymine, mutation of thymine to alanine. GTT becomes CAT. Instead of glutamine, valine comes and it leads to sickle cell anemia. Remember that sickle cell anemia is a disease where our RBC becomes sickle shaped, like very constricted. So what happens? their oxygen carrying capacity gets reduced. Understand? Oxygen carrying capacity gets reduced and it leads to anemia. So it is termed as sickle cell anemia. Okay. So SNPs can be a transition like purine to purine, that is Ag is termed as purine, Ct is termed as pyrimidine. Okay. So either it can be a change of purine to purine, pyrimidine to pyrimidine, or transversion. That is purine to pyrimidine or vice versa. Okay. And these are involved in many diseases, even the cancer also. Okay, so the research, many people do research on where the exact position, why the change has happened, where it has happened, and how to reverse it. Okay, and these are some of the databases. To all the persons who are interested, can see these links. They are giving the details of various SNPs, which are already shown. Okay. Alright, so now understand, like we are moving forward on to the next part, ESTs, express sequence tags. Understand a small concept that their length is very small, only 200 to 400 nucleotides in the DNA. So this very small length, like it's very short, it can be there anywhere. This position, this base, set of bases can be present anywhere in the genome. So if you just compare this strand on a big strand of DNA, you can find multiple locations. So it's not that much accurate. But still it is used. Reason is it is the part of cDNA. Okay, it is not DNA but cDNA. cDNA means DNA forms mRNA, mRNA forms protein in the cell. In a working body, DNA forms mRNA transcription process and mRNA forms protein translation process. So once it is formed mRNA, all the complexities are gone. It will 100% produce the same protein. So if you like a back transcribe the mRNA into a DNA, there all the complexities, all the introns, all the problems are gone. Okay, this will exactly form the same protein. So this is cDNA, which is the exact blueprint of the protein getting produced in the cell. Are you understanding? cDNA is nothing but exact blueprint of the protein getting produced in the cell. And because of which what happens, we can exactly localize the position of various genes in a chromosome. We understand that its length is less, okay, because of which we need several ESTs, okay, to map the genome properly. A few ESTs won't work out. Now, what are the limitations here? Limitations is as we discussed, it is only the partial sequence of genes. Second, because we are taking from DNA, it can be like having the contamination of various like different sequences like vector sequences coming used in the plasmid sequences. Okay, introns, ribosomal RNA, mitochondrial RNA present in a cell. So several different sequences which are meaningless to us, they also comes up. So we need to separate. So separation accuracy further decreases the overall accuracy of the process. We'll get some quantity, which may be troublesome. Okay. Lastly, the weakly expressed genes. Some genes are there in the cell which are very expressed for a very small time. So it's really difficult to get the exact mRNA for all of them. If we get mRNA, the chances how to get into cDNA, how to get their cDNA, because of all of these reasons. Okay, because of contamination by the vector. Okay, so because of all these troubles, the entire problem of getting the ESTs is substantially less. The accuracy is very less. So see, unlike ESTs, GSS includes like mapping of the various introns and exons. It considers all of them. So what is the advantage? Like we can take the GSS, map it anywhere in the genome, and we'll get the exact location. So now understand that only the problem here is the GSS is fragmented. We'll get the pieces. Like we need the entire coverage is not there. Some piece here, some piece there, some piece there, like that. So we cannot cover the entire genome. Means only GSS if you take, like you cannot completely map the entire genome using GSS. Okay. Second, like because of this trouble, we cannot know the exact order of the genes or the like what is A, B, C? Is it BCA order? Gene B, gene C, gene A comes, or gene C, gene B, gene A, gene C, gene A, gene B, or various different orders. What is the exact order? So now what is the trouble? Because of all of this, we are bound to use all of them, like ESTs, SNPs, GSS, all of them together we use just to map the genes. Okay. Alright, so now we are moving on to the second phase of this season. Here, what is the main part? We will be discussing on the gene prediction methods. Okay, and gene prediction tools used for it. The gene prediction means we have annotated, we know how to annotate the genes, but what is the exact position in the genome? Like what is the gene as such? If we have a genome, I ask you, okay, what is the gene? What is the location? Okay, what is the exact sequence of gene? Tell me the entire things. Annotation means if you have the sequence, you can know, predict where's the sequence, where's the location. You can get some idea. But if you don't have the sequence, nothing is there, then how do you do it? Okay. Or if I just give you a sequence of DNA, I ask you tell me the position of this gene in this DNA. How do you do? That is gene prediction. Okay. So the entire concept here is like based on some key parameters, you will try to predict. Okay. You will try to predict the entire part. So gene prediction is an important step in genome annotation. Also, once we have the idea of what is the gene, then only we can do the function prediction. Correct? So this is that part. And it's essential for understanding the genetic basis of disease. However, gene prediction is a complex process and is like very challenging because of presence of same like non-coding regions, the introns, okay, which are always scattered between the genes or within the genes like that. Alternative splicing. Understand that those are persons who are new. Understand that even if the exons are there, the functional key element, the coding segments are there in the gene. Okay, they need to be combined together in the transcription process to form the mRNA, which will then transcribe as a blueprint to a proteins. But like if the say suppose exons are A, B, C, D, okay, understand that in all the genes or they with all the proteins it won't produce the exact ABCD order in the proteins. It may be ABD, ACD or AD only. Means some like segments can be selectively picked or selected to form proteins. Okay, so it leads to protein variants coming up, some of which are functional, some of which are having the disease. This is termed as alternative splicing. Third is, even if the gene is there, it is a pseudogene. Means it will have the features of gene, but it is not functional. Okay. So see the basic part. If this is the DNA I give you complete, and this exact gene, the question is to find this red mark gene. That is the question. Okay. To move forward, so as we discussed earlier, annotation is means predicting the normal function of gene, mode of inheritance, genotype, and frequency of variant in a particular population. Okay, now I told you protein variants are there. Means there's some variation coming up in the functional aspects also for these variants. So annotation means we need to exactly have the idea which variant gives what change in the function. We need to have the clear idea and how much percentage is there for each of these variants in a population. Okay, so understand that when if I have the gene sequence, I'll check it in the database using the BLAST, basic local alignment search tool. Okay, I'll put it in the NCBA server and using the BLAST algorithm. Or I can use hidden Markov model, okay, to be more accurate. The, the scoring methods which are used is sequence identity, sequence similarity, e-value. Okay, this is the part which I've been telling you earlier. Exon 1, intron 1, exon 2, intron 2, exon 3. So all the exons are coding segments which are lastly producing the proteins. And introns are like base elements. When the transcription comes, it forms here. And then these are removed. See, all these introns are removed in the splicing. And only this produces the final translation. Now, cDNA means exact this part, this part, this part. The final mRNA which is produced just before the translation can be converted back to DNA. So that is the blueprint of the protein. Okay. Now, as I told you, the gene prediction method parameters. What are the parameters are? Like there's some specific key feature to recognize like exon or the intron. It's okay. So exon means exon is just like exon completes at the GT-AG part is there. See GT on this side, AG on this side of the intron. Intron starts with GT and at AG. GT, anything can be there at A or G followed by AGT. So these bases start the intron. And where the intron ends with pyrimidine. Okay, 10 bases of pyrimidine continuously. Anything can be there. Any pyrimidine can be there. 10 of them, followed by any base, then CAG. This is the end point. This is the specific regulatory part which is conserved in the intron. If I use syntax in the computer data, find all the intron boundaries using the this is termed as a regular expression in computers. If I use the regular expression of this, I can find the position of introns in the DNA. And then I can find, okay, so intermittent segments are the exons. Like that I can work out. Okay, and as because exons are combined together, okay, two exons are combined together during the splicing. Then I can call it as donor splice and acceptor splice. When intron comes in the middle, GT NAG. N is this part, anything and with the complete part. Okay. But the problem is entire human genome, like human genome having nearly billions of base pair, within which out of billions of base pair, only 3% is genes. Others, we don't know. Understand? So this is the major complexity. So God or like nature did not give us the entire 97% genome for nothing. It has some function. What it is, we don't know. And that is not the scope of this course. So I will not be discussing that. Many false splice sites are there. Means these segments are there, conserved segments of introns are there. They are not introns. I'm sorry. Okay, they are not introns. Very short exons. So if the exon is only 3 bases, it becomes difficult to recognize. So many times the gene prediction tools ignore them. But this decreases the exact accuracy. Because whatever I want will not be resulted. It results into many long, very long introns, which is not correct. Okay. Alternative splicing products cannot be resulted by this method. It's really difficult to recognize because you cannot find out which of the exons will be combined together to protein. Understand? Alright. See, this GTAG part, it is not only conserved in humans, it's present in C. elegans also. It's a very specific information. Alright, so the computational parameters which are used to predict the genes like on a DNA are like these in intron boundaries GTAG, remember the part. These are very basic signals which are used to predict the genes. GenScan was the first tool to do it. Okay. Besides that, the regulatory elements, the signals which are matches to like transcription factor binding sites, these are specific key segments which are always bound by the transcription factors. Okay, so we can search these signals. Okay, or we can use the interspecies alignments. That is, take several sequences belonging to different species and then compare all of them. Okay, using an alignment as we discussed earlier. Using an alignment, we take several sequences and then we can work out. Okay, so whichever is the conserved portion, we can designate the part and work out. So system exactly do the same thing. Okay. So see, there are three methods of gene finding. First is statistical or ab initio method. These basically use the GTAG boundaries, basic signal sequences, just compare them in the DNA and then find the gene boundaries. So the accuracy is very less. Second method is the comparative method. Here we take several DNA, several species genes and then compare them together using the alignment. Here we believe that exons are more conserved. So if we know them their boundaries, we can exactly find the position of genes in the different DNA. Okay, different species. Third one, homology method use the information of proteins to find out the location of genes in DNA. We go reverse way. Okay, using homology. Okay. Now understand that what is the gene prediction in prokaryotes. What is the method? The gene prediction in prokaryotes only uses the information which is the known structure of genes, which is the standard known structure of genes. That is, the promoter RNA complex binds here at the starting of gene. And starting of gene is defined by like -10, -35 bases. Starting of gene. These are conserved segments here. Ribno box and Gilbert box. T8080, also termed as Tata box, or TTGACA. If I know these positions on a DNA, I'll know that somewhere down the side, at these distances, the gene is there. Another peculiar specific feature is the untranslated region, the conserved gene segment which is not coming up in the mRNA or the protein. And then the gene starts. The starting is start codon and the ending is stop codon. Okay. So this is the ORF, open reading frame. Understand? Now understand that as because there are three reading frames here. Okay. Like there are two double-stranded DNA. Double-stranded DNA is the two strands are there. So there can be three reading frames for each amino acid. Okay, you consider triplets at a time. Okay, take all three together. Okay, then leave one, and then take the next three, then leave two, take the next three. This gives you forward. Same way you will get the reverse. Okay, then check out what are the start codons. Okay, ATG is there, and you will get the stop codons. So we can check for these start or stop codons. Understand? So when you are checking these, you see that start codon is there and stop codon is there. So this is not the gene. Not even a single base amino acid is there. Okay. So such kind of things leads to errors. But we still consider to predict the genes. Okay. Okay, so like till now we have studied that the gene prediction is a critical step in the annotation of the genome. And we have used several methods, three category of methods to predict the genes. Now we'll be moving on to the further details. That is gene prediction method, gene prediction tools 2 in the next phase of this session. Okay. Here we will be discussing about ORFs. Okay. So ORFs, like understand that open reading frame. If I just check out, understand the trouble here is I shown you start codon TAG. What is the probability of T on the DNA? There are four bases. To get T anywhere is 1 by 4. So what is the combined probability of TAG on a genome? 1 by 4 raised to 3 means 1 by 64. Okay. So 1 by 64 means after every 64 bases there can be one TAG. So now you understand if I just check by positions, the probability does not work out, it fails. So means still we know better methods, better computational strategies on more diverse information to predict the genes. Okay. So even if you can use tocodone usage, okay, this works better. Alright, so students now see like the gene prediction in eukaryotes, how it is done. Okay, so now in eukaryotes, what is the trouble? Introns are there, like internal exons, terminal in exons, initial exons, and we need to combine all of them. What is the signal? GTH signal, and the start codon and the stop codon. along with the like 5' untranslated region, 3' untranslated region. These information we have. Okay. So here also see that GTG part is conserved. Okay. And this is the alignment part. Position specific scoring matrix. If I align all the amino acids, I build up a matrix of amino acids like this. So as per the positions, I can see that some amino acids are particularly conserved. See, what are the programs here? Like either the GenScan is there, see, which was already there. Here it used modified HMMs now. Okay, like this way it has been improved. Okay, by using HMM, by using the gene structure in more details. The twin scan which is there. Okay, use both HMMs and now similarity. Okay, so it considers the alignment along with the basic principles and evaluates them using the hidden Markov models. Comparative gene finding. This is the second method now. Comparative gene finding implies like building up the alignments and finding the similarities. Okay, so here the program is Rosetta. It first compute a global alignment of all the homologous sequences, at least two of them, and then it finds the like conserved regions. Okay. So if a problem is given a known gene, an unannotated genome sequence, find the substrings of genomic sequence whose concatenation, when I link them together, it should give me the desired gene. That is what it means. Okay. So these are the like, small segments. I'm calling them as islands. Islands of segments on a big genome, so that if I combine them together, I'll get the desired gene. We are not discussing about the function. We are only telling the gene extraction, gene finding, gene prediction. Okay, our objective is to designate the particular gene on a genome. Okay, annotation means finding its function. Okay, so what are the limitations here? Okay, these the tools like twins can use similarity also. So GenScan, what is the limitation? It does not use similarity. So TwinScan is of course more accurate. Okay, like as because TwinScan or tools like using similarity are able to predict the variants of genes also by checking different proteins coming up from the same gene. But in the case of GenScan, we cannot predict all this. Okay. So next tool is the genome scan. It incorporates the similarity information also in the GenScan. Predicts gene structure which corresponds to maximum probability conditional on similarity annotation, similarity information. Okay. So it considers not only probability of an HMM base, it considers the similarity. So now it summarizes to like several different things have been developed so far, and I can categorize all of them in these order. Initially, understand that genomics, genome DNA like sequencing was done out, so people started using all the sequencing strategies to work out. Okay, so database size has drastically increased not only in genome database but in protein database. Okay, if you compare NCBI gene database, NCBI protein database, you will find billions of sequences. Okay, then we started building mRNAs using microarrays and other things, and getting the serial expression of gene, serial analysis of gene expression, SAGE method. Okay, we started finding out what are the mRNAs getting expressed. We could build cDNA. So these libraries started coming. Then the proteomics took over. We realized that this gene or the mRNA is not enough. We require protein to get the infer the function, know the functional details. Okay, so this protein function is much more important. So getting the functional aspects, like we started working out on protein structures, like quantitative profiling, protein cataloging, logging, these kind of techniques. Understand? So now, like putting up all together in the computational boom era. Now the things are changing. We have started like finding out the catalytic activity. Okay, what is the protein? What part of the protein is working? What part of that enzyme is working? What are the amino acids involved? What is the mechanism? What is the special like proper subcellular location? Which organelle it is working? Under what conditions it is working? If it is working, like what are the modifications it is going on? Okay, so modifications are there means post-transcriptional or post-translational modifications, all of them are going on and are changing the function. We need to have the clear idea about everything. Okay, because it will change the protein expression or the protein function. So how with time, when the protein was produced, the protein function kept on changing or its location kept on changing. Okay, so protein phosphorylation, okay, different acetylation of the different groups which are taking place. Okay, we normally add the functional groups on the protein to label them, to change or like make them more active. Likewise, protein-protein interaction maps, because protein is not alone, it works with multiple proteins. Okay, so protein works in function as a network. One protein gives signal to other proteins, and then this signal pass on further to do the desired function. So this is protein-protein interaction map. Okay, so if multiple proteins are involved, like in a human body, 30,000 proteins are involved, but all 30,000 do not work under the same condition. Okay, so if I take glucose, if I take milk or tea, only a few proteins work. Okay, so the body sense the glucose or lactose or tea or milk is coming. Accordingly, the things work out. Okay, so for sugar, some other enzyme, for milk, some other enzyme, likewise for tea, and then the body works. So body is specially passing on the information in a channel. So this protein-protein interaction map is very crucial for the body to function, and it involves not only proteins but other molecules also. Understand? I just gave you the example of glucose or the like milk. So along with protein, milk is also involved, and milk is not DNA, RNA, or protein, no biomolecule. It is additional molecule. So it is additional component also involved here. Okay. So I'm calling it as system components. If I'm calling the body as a complete system, the body has multiple proteins, multiple carbohydrates, multiple lipids, and different additional carb like anions, cations, different energy like producers, like that, and thousands of them. So if I know the exact concentration, exact thing at every place, and how it is changing with time, like what are the things changing, what are the properties, I can find out what are the key constraints which are changing it. Although by saying it is simple, but it's actually very difficult to find. And this is a part of systems biology, where all of them are involved, where we analyze the complete network together. Understand? Okay. Now I'm moving to the last part of this session. That is the molecular predictions which are feasible for the. So see, in this next slide, what I mean to say is I've shown some organisms along with the proteins, and this graph shows how many of them have been characterized using four features. Something characterized experimentally, which experimental validation has been done that this is the same function. Characterized by similarity. Experimental validation is not done. We just know them by functions like similar. Because they are similar to some other gene in the some other species where it is already checked experimentally. Okay. Unknown but conserved. Okay. Conserved gene is there, but the function is not known still now. Reason is it is something else. Whatever it is seemed to be by similarity is not the same. And unknown, no similarity at all. These are very troublesome. So now if you know, now if you see here, characterized experimentally or characterized by similarity is the largest number here. And unknown conserved, unknown no similarity at all is on the lower side. So similarity is useful, but it does not always help. Does not always guarantee that whatever it is seems to be, it is exactly the same function. It can be something else. So how to figure it out? You need to determine it experimentally. You need to validate it experimentally. Then only we can be confirmed. Okay. So to figure it out, understand that annotation methods are defined using 7 different strategies. We use 7 different strategic functions to define annotation. First is homology. Okay, sequence similarity is there, and a significant sequence similarity, at least 30%. Okay, besides that, we do sequence comparison. Okay, then the sequence features and motifs. Whatever the gene motifs which are present in one species should be similar to the ones present in the other species. Okay, subcellular localization means whichever the organelle it is working in one species, the similar organelle it is expected to be same organelle it is expected to be working in other species also. And all of these, are the characteristic features considered by gene ontology strategy also. There's a go strategy there. If you put any gene, you will get the all this information. Okay. Structural databases. Many times understand that all these features do not work. Still the protein is something new. Then what to do? So we consider the protein structural database. That okay, sequence is not there for this gene, this protein. No such similar sequence is there, but is a similar protein structure existing in the protein structure database (PDB). And if it is there, I can compare my sequence with that structure to then correlate the function. Okay, metabolism database. Okay, this is another part like your metabolic activity can be predicted by using the key features. Interaction database, there it may have the idea. Okay, so these are different categories of annotation methods which are used. Then, how do you work it out? Where I should have the more confidence. So if I use with the transcript, that is one part. Protein, another part. Sequence conservation is just one element. Proteomic data is just one entity. Transcriptome data, see? Okay, it's just another entity. So now if I ask you here, like if you have only one of them, will the gene prediction algorithm be confident that you will be confident that this prediction is correct 100%. Yes or no? I hope you will be all thinking like no only. Reason is these are prediction methods. Okay, they may be right, they may be wrong. But if you take more algorithms, okay, transcripts, protein, conservation data, proteome data, transcriptome data, everything. But then also protein function is just found to be one or two of them. Then that implies these are most likely functions there. And then you can determine the same functions experimentally. Only one or two of them that you can confirm. If it is found to be, the experimental blind game is minimized. Okay, you need not check every single function for these proteins. Okay, so that is the main advantage. So if you check just one strategy at a time, you may get 10 different functions. Then when you put the second layer, the search minimizes to few. Third filter, third algorithm. We are making our algorithm more and more robust, more and more correct. Okay, so the chance of inaccuracy for missing all of them together is very less. That is the entire game of probability here. Okay, so in all such things, we are always putting the multiple different data put together so that our chance of accuracy increases. Okay, the rankings will be done. Understand? Whenever I'm putting the algorithm strategy, I'm putting the ranking. Is it right or wrong? Correct. So is it right or wrong? So whenever I'm checking each and every part, each and every score on every single data, I assign a score. Is it right or wrong? Okay. So based on that score, I can find out the rank. Which one is more correct? Okay. So some strategy which is consistently correct, most of the times it is correct. Then I can use it 100% for a blind run when I don't know anything. So the similar percentage of accuracy I'll expect there also. Okay. So to boost the accuracy of genome annotation, I use an integrated approach. I use the gene predictions. Okay. All of them. See, now I'm summarizing all of them this way. It's in the single integrated approach, which is summing up the entire part to put increase the overall accuracy of annotation. Consider the gene predictions, comparative alignments using BLAST or HMM, then transcriptome data, proteome data. I always use the promoter element database as an additional filter. Also update the score for data for proteomics, transcriptional, and the translational data. This allows me like as an additional filter to improve the accuracy. I'll also consider the crisscross conservations. That if some crisscross conservations are there in different species, I consider them because some sequences may be bigger, some may be smaller, but I still may have the conserved sequence segment. These are crisscross. Because the conserved element is now shifted a bit distant. Okay. I'll locate the missing exons or missing introns. Then I'll find out alternative splice variant. It's because they lead to wrong annotation and I should be carefully understanding them. I should properly understand that there can be multiple variants of the same protein. So I need to consider that probability also. Then once I am confirmed that this is 100% alternative splice variant, multiple proteins coming out from the same gene, I can have the idea because I know exons, I know proteins, I can easily find out. Okay, this is just an example. One protein coding gene is like that, see, in one species. Another species is broken down into these only small protein. See, other one is a bigger protein. 1, 2, 3, 4, 5, 6, 7. See, next one is very small. Other one it's very small, only 4 exons. See, here it is 6 exons. But the length of the exon you can see here. It is substantially different in all of them. Are you understanding? But when I am comparing all of them, this part is not overlapping with this one. But still I know it is here. Okay. This is the advantage of putting all of them together. And all this increases the overall accuracy. Okay. So gene annotation algorithm, see. I use BLASTP or SciBLAST tools. I use SwissProt because SwissProt is manually annotated. I use the HMM filter. I use the protein family database. I work it out. And all this is by part of gene ontology, which predicts molecular function, biological process, cellular component. Okay. Gives me the notation three different things: function, cellular component, and the biological process where it is involved in. Okay. And these are the functional resources which are there. See? All of them are like different parts of the protein function. Some are giving like signaling site. See? Some are giving like a protein information resource, protein network database, protein like a protein functional element site, gene ontology, like an interconnected network which is there, like Proto Encyclopedia of Genes and Genomes, TIGR fam, protein fam, okay, DDBJ, EMBL GenBank, okay. So what are the problems in the protein annotation lastly, or the assignments? If somebody do a wrong annotation earlier, 5 years back, what will happen? My new algorithm, when I'm checking the function, it will check the function against that old sequence. So if my sequence is say 80% similar to that old sequence, which was already wrongly annotated, my algorithm will tell that it is having the same function. Correct? It will guarantee that this is having the same function. But the problem is that the base was wrong. That old sequence was wrongly annotated. So this means the error done earlier is now propagating, now moving forward in the new genome as well, new predictions as well. This is the biggest problem in all these annotation methods. Because of this only, the Swiss plot has been developed, where the manual annotation is worked out. Researchers use all these discussed strategies to annotate a gene, and then only they confirm that this function is correct. Otherwise, they change it. Okay. So, what are the function prediction, the problem areas? Analysis of proteins with low sequence complexity means some repetitions are there. Okay. Then identification of domains in the absence of close homologue. When you don't have any close homologues, how do you find out the protein islands, conserved islands, protein functions? Finding the possible functions, evolutionary distant paralogs. Okay. Identification of protein coding regions. How do you find exons reliably under all these conditions? Okay. What it leads to is that for my gene, if it is having conservation at multiple parts, I can say it as one protein like is having multiple functions. Okay. Or it is a single protein having multiple different small segments. See? It is functionally diverse, or it is highly divergent family with a single function. Multiple different proteins are there, but it's highly divergent family. But it is having a single function. Sequence similarity may not be there, but functionality is conserved. Okay, so this is a big challenge. So if I do manual error, manual annotation, these errors I can resolve. Incorrect exon boundaries, merged or split missing exons, missing UTRs, missing alternative splice variants, degenerate transposon, okay, that get annotated as protein coding gene by an automated algorithm. Okay, this normally happens. Recent study has shown that the results of various tools are not always identical. I have shown you multiple features are involved, and different algorithms prioritize different scoring strategy. Okay, it's difficult to put all of them together consistently always. Because of this, variation comes up in the score. Okay. No current tool yields a biologically correct annotation for every gene. Because for every gene, you don't get all the strategic parameters. Okay. So that putative proteins, it leads to two terms. Some is putative, which is sharing a new gene is sharing a high similarity with a known gene. Okay. Here it is not functionally characterized. So I'm calling it as putative. It should have likely have to have this function. Hypothetical protein is it's a protein coding gene, but no experimental information is there. But it should be a protein coding gene. Okay, like that. This is a recent paper, 2018 paper, where whatever the terms we have discussed, they have used all of them and then shown that they have annotated 13,413 genes. They changed the annotation. Whatever the earlier function was there was found to be wrong. So they corrected the function. Okay. And then shown that if you use all of these parameters, transcriptome data, homology based prediction, de novo prediction, using several different scoring methods, okay, we can annotate the genes better way. Okay, this is the most important part. So they could annotate 13,413 genes. So this is the biggest part. Okay, see. So the understand that whatever the methods or whatever the parameters we have studied in this lecture are constantly being used. Okay. Thank you so much for your time.